You're at the cutting edge of cardiovascular sciences. I'm your host, Professor John Cook at Houston Methodist Hospital, and I'm joined in the studio here, DeBakey studio, by my junior faculty member, Dr. Lee Lai, who is going to introduce a very special guest to us who is really the king of angiogenesis, or the baron of angiogenesis. Please.
Good morning, everyone. Today it's my great pleasure to introduce Professor Peter Camilliet, who is the head of Laboratory of Angiogenesis and Vascular Metabolism and former director of the VIB KU Leuven Center of Cancer Biology in Belgium. He is undoubtedly the leading figure in angiogenesis research, whose work has inspired a lot of researchers, including me, in this field. Professor Camilliet got his residency in internal medicine in 1984 and a PhD in 1989, both from University Hospital Leuven, Belgium. He did his postdoc training at Harvard Medical School and then at Whitehead Institute, MIT. After that, he joined his home school, the University of Leuven, Belgium, and did a lot of very fundamental research to identify the molecular and cellular basis of angiogenesis in health and disease, and in particular the role of vascular metabolism and vascular heterogeneity herein to ultimately identify novel therapeutic pro- or anti-angiogenic strategies. Professor Camilliet's exceptional contributions to the research and the clinical applications were recognized by a lot of international and national awards, including Franke Prize, which is a prestigious Belgian scientist prize in biological and medical sciences 2002, and the Ernst Young Prize in Medicine, which is one of its highest endowed medical awards in Europe in 2010. Yes. He also got the Heineke Prize for Medicine, which is bestowed by the Royal Netherlands Academy of Arts and Sciences in 2018. In 2015, King Philip of Belgium granted him the Nobel title of Baron. He had elected as a member of the International Academy of Cardiovascular Sciences and international honorary member of the prestigious American Academy of Arts and Sciences last year. So today it's our pleasure to hear from Professor Camilliet about his insights into novel anti- and pro-angiogenic targets in endothelial metabolome and the newly identified immunomodulatory endothelial cells as possible new targets for alternative immunotherapy in cancers.
Welcome, Professor Camilliet, to the show.
Hi, how are you? Thank you for the invitation.
You, as Lily just told us, you have been a leader in angiogenesis, and angiogenesis plays an important role in cancer biology, but also in cardiovascular disease. And let's talk about that for a moment, because most of your talk is going to be about the angiogenesis in cancer biology, but it's directly applicable to plaque neovascularization, for example. Tell us what your thoughts are about plaque neovascularization and its role in atherosclerosis.
Yeah, that's very important. It's not so well studied because, in part because in the mouse you don't have really large plaques, and so there is not so much or little to non neovascularization. So we lack the animal models to actually that are suited to study this process. But obviously it's very essential. Also in aneurysms, etc., you see that there is increased angiogenesis. With the vasa vasorum and inside the plaque.
Mm-hmm. Do you think that it's possible that anti-angiogenic therapies could be directed toward plaque neovascularization and for the treatment of atherosclerosis?
Yeah, I don't see no reason why not.
Yeah,
it's a question of finding the right targets.
Yeah. That being said, we do know that anti-angiogenic therapies can have adverse effects on the cardiovascular system. So cancer anti-angiogenic therapies have adverse effects on the cardiovascular system. Let's talk about that for a moment.
Yeah, but that depends on the dose also of let's say, if you give anti-VEGF or VEGF blockers, the healthy endothelium also needs some residual levels of VEGF just to stay healthy and to survive. So if you block this completely, then you cause all these endothelial cell dysfunction and these side effects. But what they do in cancer patients is to not completely block the VEGF levels. in certain patients that will happen, and then they suffer these endothelial cell dysfunction related side effects such as thrombosis, sometimes bleeding, barrier disruption, etc. So that's a challenge to dose it and to personalize it for each patient, because we lack actually good biomarkers for such treatment.
Yeah, well, that's a good point. The biomarkers for angiogenesis. Are you doing any work in that area to understand the role of angiogenesis, or at least the utility of biomarkers for angiogenesis, either imaging biomarkers or circulating biomarkers for pathological angiogenesis, such as that which may occur in cancer or plaque?
No, we're not doing any imaging, but actually we get some novel insights from the single cell studies. And I will show during my talk about the presence of novel endothelial cell phenotypes in breast cancer, for instance, and they might actually be good biomarkers.
Interesting. Well, we're going to roll your pre-taped talk in just a moment, and then we'll have some questions afterwards. You'll be joining us for the question-answer session,
Absolutely.
And that will be wonderful. But before we leave, I just want to ask you a question. You've gotten a lot of awards for your work in angiogenesis, but I'm intrigued by the award that was bestowed upon you by King Philip, the title of Baron. And tell us about that. Tell us about, did he invite you to the palace to bestow this award? Tell us what that was like.
Yeah, well, first of all, they call you personally. Mm-hmm. So I was called on a Monday at 6 p.m. and the palace asked, do you want to accept this honor? And the reason they gave was actually something that I could not refuse, and that is that they wanted to promote academic research for the junior scientists. By giving or bestowing a Baron title to a leading scientist. And then you're invited together with other people at the palace, and you meet and greet the king, and you have a small chat, and you accept a charter basically, and that's it.
What did the king say to you?
He's a very friendly person. Accidentally, yesterday, so you know about the World Championship for soccer.
You'll have to tell me. You have to tell me about it. I follow baseball.
So it's starting next week in Qatar, and so the Belgian soccer team has been doing very well in the previous rounds. This time, I don't know whether they will succeed, but to support them, actually, he made a small movie. Which was very hilarious and was extremely well done. It's like a 1 minute show, and it was on national television, and he got a lot of, great applause for it. He's very kind. Officially he's a king, but when you talk to him, he's just a normal person.
Oh, wonderful. Well, I think we'll go now to your talk, which was pre-recorded, and then afterwards we're going to have a Q&A with Professor and Baron Carmelite.
Okay.
Thank you very much for the opportunity to discuss the role of endothelial cell metabolism in heterogeneity. One of the key problems for the angiogenesis field today is shown on this slide. Here you see the progression-free survival of cancer patients treated with bevacizumab, which is the antibody against VEGF. But this benefit is usually lost when we look at overall survival. We know now that this is due to intrinsic refractoriness and acquired resistance. At the same time, we also know that endothelial cells are heterogeneous, yet tumors are indiscriminatively treated with anti-VEGF. So how can we overcome the resistance to anti-angiogenic therapy? Well, we focus on endothelial cell metabolism and heterogeneity. On this slide, you see one of the mechanisms of resistance against VEGF-targeted therapy. So when an endothelial cell is stimulated by VEGF, it induces sprouting and angiogenesis. When you block VEGF, then you block angiogenesis. But in a cancer, there are many additional growth factors that actually can lead to escape of angiogenesis. And blocking each of them separately will only make the tumor cells smarter and lead to more and more of these angiogenic growth factors. So some 10 years ago, we decided that we needed to hit the endothelial cells in their very heart. And we thought that this could be metabolism onto which these growth factors converge. So the concept was blocking the engine rather than the drivers. We initially started with reverse metabolomics, whereby we study a particular metabolic pathway in vitro and also prove that it has an angiogenic activity in vivo and show therapeutic proof of concept. We now also, since a few years, have focused on forward metabolomics, whereby we isolate endothelial cells from mice or patients to multi-omics profile and identify their heterogeneity and eventually metabolic maladaptation. Before showing you some data, I want to share with you some of the tools that we developed. The EndoDB is an endothelial cell transcriptomic database that is interactive. We also developed Biomex, which is a suite of more than 250 software programs that allow non-expert, non-bioinformatics expert wet lab scientists to actually do their own bioinformatics analysis quite rapidly. And we also developed an endothelial cell genome scale metabolic model, which is in fact a computational model that allows you to predict which metabolic pathways are active and essential in endothelial cells. So let's look at a few examples. So we first targeted lipid metabolism in endothelial cells and focused on CPT1A because this is a rate controlling enzyme. And so we found actually a phenotype in the differentiation of venous endothelial cells in red here to lymphatic endothelial cells in purple. And this, according to the traditional model, occurs, it's based on the master transcription factor PROX1, which you will see in a second, binds to the promoter of VEGF receptor 3, as well as several other critical lymphatic markers that induce then the lymphatic vessel development. Our findings actually show an adapted model of lymphatic development where the purple lymphatic endothelial cells still transdifferentiate from the red venous cells, and whereby PROX is still essential. But PROX is sort of a very smart transcription factor because it hijacks metabolism and in fact fatty acid metabolism because it binds to the promoter of CPT1A, which leads to an increased fatty acid oxidation that leads to the generation and production of acetyl-CoA, which is then used by the acetyltransferase P300 to actually enhance the expression of VEGF receptor 3. P300 binds preferentially to these lymphatic promoters by forming a bond with PROX1. But we also showed that proliferating endothelial cells have a high fatty acid oxidation, which leads to the production of acetyl-CoA, which in conjunction with other anaplerotic substrates sustains the TCA cycle for the production of nucleotides. And so when you block CPT1A, you see a reduction of the DNTPs and impaired proliferation. And when you block CPT1A in vivo and you treat mice with them, you can see then in an ocular model of pathological angiogenesis that FAO blockade reduces pathological angiogenesis. Now, fatty acid oxidation also controls nucleotide synthesis in lymphatic endothelial cells. And ketone bodies are another source of acetyl-CoA. So that's why we focused also on ketone body metabolism, and in particular on OXCT1, because it's the rate controlling enzyme. And so when you silence OXCT1, you see in vitro reduced vessel sprouting and lymphatic endothelial cell proliferation. And similar like the fatty acid oxidation, ketone body metabolism in lymphatic endothelial cells produces acetyl-CoA to sustain the TCA cycle for DNA synthesis. And when we then silence or analyze lymphangiogenesis in mice that lack OXCT1 in lymphatic endothelial cells, then you see here that there are fewer of these green lymphatic vessels in vivo. The question then arose, does the ketogenic diet improve lymphangiogenesis? And yes, indeed, when you treat wild-type mice and give them a ketogenic diet, then you increase lymphangiogenesis, the vessels here in green. So the question arises then, can we give dietary ketone bodies, which is a source of acetyl-CoA, to treat lymphedema patients? Indeed, normally the lymph is actually drained by lymphatics into the venous circulations. But when breast cancer patients undergo surgical resection of lymph nodes, actually this circulation and drainage is impaired and lymphedema may occur. There is absolutely no cure and only symptomatic treatment available so far. We then focused also on endothelial heterogeneity. And as you see in this movie, VEGF induces the formation of a vessel sprout. And at the tip is in blue here is the tip cell, which leads and guides the vessel sprout. Behind are the proliferating stalk cells. But you will see in a second that the blue tip cell actually loses its competitiveness to stay at the tip and is overtaken by a stalk cell that now becomes the tip cell, so that always the fittest and metabolically fittest endothelial cell leads the vessel sprout. But an important conclusion is that endothelial cells have no fixed fate, but can dynamically interchange phenotypes. So endothelial cells are highly plastic cells, but how heterogeneous are they? So we first studied this in human lung tumors, and as you can see here, you actually have blue venous endothelial cells, then you have purple immature and orange tip cells and black proliferating cells. You also have red arterial endothelial cells, and then you also have these green capillary endothelial cells, and they're all statistically separable. And what you see in a tumor, so the NEC stands for normal endothelial cell, TEC for tumor endothelial cell, is that in tumors you have many fewer capillary endothelial cells, but you have more of these angiogenic immature and tip cells. And surprisingly also you see enlargement of the venous pool, which will become interesting for the later discussion. Remarkably, however, there are less than 10% of proliferating and migrating tip cells, and these are the presumed angiogenic targets of anti-VEGF therapy, and it raises thus the question whether this small fraction may contribute to resistance against anti-VEGF. So most tumor endothelial cells have actually non-angiogenic phenotypes, and this may cause actually a paradigm shift in our thinking. We should maybe not only prune tumor endothelial cells, but also retune tumor endothelial cells, and I will come back to that later. For instance, here we also noticed that a fraction of these endothelial cells have actually immunomodulatory activities. For instance, some are semi-professional antigen-presenting cells. Others have a gene signature involved in microbial defense or in scavenging. What happens in tumors is that these immunomodulatory endothelial cells are actually suppressed in number. But at the same time, what also happens is that at other locations, probably you have an increase in high endothelial venule biogenesis, which can lead to T-cell recruitment. So on one side, you have immunosuppression, and on the other side, you have immunostimulation. In any case, TECs are the first-line defense system encountered by immune cells and may thus be more important than previously thought for developing new immunotherapy targeting these cells. In fact, we think that they're so interesting that we wrote recently a review about these immunomodulatory endothelial cells. And this is just an illustration how actually we reviewed their activity and numbers in a tissue-specific manner, here, for instance, in the lung. We also discovered the previously unknown breach and pre-breach endothelial cell, which are involved in podocyte rosette formation in preparation of tip cell migration, and might also be considered therapeutic target. We studied the differential sensitivity to anti-VEGF, and compared to the control, we used blockers and antibodies against VEGF receptor 2. And what you see is that actually tip and breach TECs are most sensitive, proliferating TECs are not more sensitive, but vein TECs are actually less sensitive, and this may have implications for the following model. In a traditional modeling, the tip cell arouses and sprouts from the venous endothelial cells, and then is followed by a stalk cell to then merge with a neighboring vessel and then become resident quiescent phalanx cells. But what we noticed in the venous endothelial cell cluster, that was a gene signature of resident endothelial stem cells. And when you do lineage tracing via pseudoderm, then you see that the sprout actually, the vessel sprouts starting from the blue endothelial cells over the purple immature and orange tip cell to then become a quiescent arterial cell. And so we would like to adapt the previous model, the traditional modeling, and propose that there, in the venous pool, there might be a smaller fraction of endothelial stem cells that first expand and then actually differentiate over varying, transitioning, and immature endothelial cell phenotypes into tip cells and angiogenic endothelial cells. And interestingly, when you block VEGF, then actually you increase this pool of the venous endothelial cells containing these resident endothelial cells, raising the question whether this is a new resistance mechanism against anti-VEGF. Now, a big challenge for the single-cell RNA field is how can we prioritize single-cell RNA sequencing targets for drug development? We thought that conserved markers are likely to be functionally more relevant and therefore used integrated multi-omics analysis of endothelial cells across species, tissues, and diseases. And so when you look at the gene expression signature in angiogenic endothelial cells across diseases, in tumors and in eye disease, then you see that they're remarkably conserved, raising the question whether there are similar mechanisms of vessel formation across these different diseases. And I will come back to that later. We also see that when we do a meta-analysis that only tip cells are actually congruent in angiogenic diseases. Here you see indeed that the breach in the tip cells are the only ones that are conserved in human, freshly isolated and cultured tumor endothelial cells and in freshly isolated murine tumor endothelial cells. And this is also true across diseases, for instance, in both CNV, dus the eye disease, as well as in tumors, tip and breach cells clustered together. So tip and breach tags are conserved across species, diseases, models, and tissues. Are tip cell markers then the next generation anti-angiogenic drug targets? Well, I'm going to give you one example here, lataxin, which is one of those high top-ranking tip cell genes that is new. You see that by immunostaining, it's really expressed indetectable in the tip. And when you silence it, you see that the green silenced cells actually are not at the forefront, not at the tip position, but trail behind. So this is a typical phenotype and characteristic of a tip cell gene. We also explored whether tags are metabolically heterogeneous, and by focusing only on metabolic genes. And so what you see is that both in the tumor as well as in eye disease, each of these different endothelial cell clusters can actually be differentiated and distinguished by a different metabolic gene signature. And what was surprising is that not only glycolysis on the top was increased, as we knew already from the bulk analysis, but now also that actually OXPHOS genes were upregulated, which is sort of surprising since we knew that the endothelial cells are highly glycolytic. Certain tumor endothelial cells, such as those in the veins, are in prostaglandin metabolism, possibly for vasoregulation and vessel sprouting, while capillary tags are involved in lipid metabolism and angiogenic tags more in extracellular matrix biosynthesis necessary for vessel sprouting. I showed you already that with pseudotime you can do an in silico analysis of how the vessels sprout, and you can see here that each of these phases is actually characterized by specific markers. But this is also true for the metabolic markers. And you see that at each of these phases, they have different gene signatures, metabolic gene signatures, suggesting or raising the question whether metabolic plasticity underlies heterogeneous endothelial cell differentiation during vessel sprouting. Now, we thought that we need to include more stringent selection criteria to prioritize new angiogenic targets because transcript levels do not necessarily reflect metabolic fluxes. And therefore, we need additional predictive methods for prioritization. And that's why we constructed an EC-tailored genome scale metabolic model, which is actually nothing less than a computational package that allows you to predict which metabolic genes are essential for certain processes, such as, for instance, biomass production or collagen synthesis. And you tailor them to endothelial cells by including multi-omics data that you obtain in endothelial cells into this computational package. And so we used an integrated multi-omics meta-analysis approach to discover novel angiogenic targets. So first we used human freshly isolated and human cultured endothelial cells and combined it with mouse freshly isolated taxonomy, then performed a Jaggard similarity to identify that only tip cells actually are conserved across patients, species, and models. And we identified already that plot one, which is one of the genes that encoding collagen and collagen modifying enzymes was highly upregulated in the top ranking position. We further completed this with a meta-analysis of transcriptomics in bulk transcriptomics in 13 lung patients, 5 different tumor types, as well as proteomics, bulk to proteomics in 93 patients of tumors, lung tumors and other tumor types. And in each of these cases, we observed that the plot1 and 2 and LOXL2L2, which are the genes involved in collagen synthesis and modification, were ranking very highly up. Using the GEM, we also identified that actually the plots are top ranked by this computational program. So we decided to silence these enzymes by treating the mice, wild-type mice with plot blocker, and as you can see, this reduces ocular neovascularization. So it is possible to actually use a multi-omics and meta-analysis approach to discover novel angiogenic targets from single-cell studies. Now, another fundamental question is how general is the endothelial cell heterogeneity in other tumors? And that's why we focused on breast cancer, where we studied early stage ER positive and HER2 negative breast cancers. And so, like in the lung tumors, we see indeed the same type of endothelial cell clusters. And on the right, you see that the activated and angiogenic endothelial cells in the tumors are actually more present than in the non-tumor part. And again, like in the eye and in the lung cancer, we see that in the breast cancer, the angiogenic endothelial cells actually cluster together on a similarity analysis. And on the right, you also see that the lung and breast tumor endothelial cells share many angiogenic targets, raising again the question whether these mechanisms of vessel formation are similar across diseases and across tissues. In this case, in the breast tumor, we saw that the vein endothelial cells are actually mostly immunomodulatory, and in the tumor, that these vein endothelial cells actually become immunosuppressive. They lower their expression of antigen presentation, of cytokines, chemokines, and vascular inflammation genes. And angiogenic endothelial cells express TGF-beta, which is involved in immunosuppression. In addition, a receptor ligand interactome analysis predicts that venous endothelial cells may regulate natural killer cells via CLEC2B and KLRF1. And this is confirmed also by immunostaining. And when you silence CLEC-B2 in endothelial cells in vitro, it impairs natural killer cell activation. But we also found a surprise, and perhaps an endothelial cell that is unique to the breast tissue. So what we noticed is that the many genes involved in lipid metabolism were actually downregulated in the TEX and thus upregulated in the normal endothelial cells. And there was one particular capillary, type 2, which we call the lipid metabolism processing endothelial cells, LIPEX, because they express many genes involved in lipid glycerol uptake, fatty acid binding, and transport lipid droplet turnover, and so on. And so they are underrepresented in breast cancer, as you can also see here. These LIPEX actually express high levels of PPAR gamma, which is a master regulator of lipid metabolism, both at the mRNA as well as at the protein level. And so it raises the questions, do lipids impair breast or prevent cancer development? And in order to indirectly address that, we actually did a retrospective study on 5,000 patients, breast cancer patients, and followed them up for over 14 years. And amongst those, a number were actually diabetic patients who were treated with metformin, which is actually a PPAR gamma agonist. And if you then look at the breast cancer specific survival or distant relapse-free interval, then you see that there is clearly a benefit when these patients are treated with PPAR gamma agonist or metformin. The magnitude of this therapeutic effect may seem small to you, but is in fact similar to that of standard of care anti-hormonal treatment in this stage of the disease. And interestingly, in these patients treated with metformin, there were also a higher number of LIPEX present. So it raises the questions whether LIPEX are a new therapeutic target for breast cancer. So far, I've spoken to you about sprouting angiogenesis, but there is also another type of vessel formation, and that's vessel cooption, whereby tumor cells co-opt pre-existing vessels. This is interesting because it is actually contributing to the resistance against anti-VEGF. We used several models. One of them was actually whereby we injected Renka tumor cells and then followed them up for the first 21 days when the tumor grows, as you can see on the right side, but they actually grow via angiogenic metastasis. When you then start treatment with sunitinib, the tumor size decreases, but then relapses, and when the tumor regrows, then actually it regrows primarily via vessel cooption, as you can see. And so the question raises, can we target co-opted tumor endothelial cells? Our hypothesis was that these co-opted tumor endothelial cells would be activated, but what is their gene signature? So here is then the representation of the endothelial cell clusters in the healthy lung, and if you compare it to the D21 untreated tumor, you clearly see in blue that you have angiogenic and also the first gray immature angiogenic endothelial cells. But if you compare it to the day 36 sunitinib treatment, when the tumors now grow via vessel cooption, then actually the co-opted endothelial cells largely resemble quiescent endothelial cells in terms of their gene signature, and there is only more of these immature endothelial cells. Also, if you compare it to real healthy endothelial cells, then you see that both are actually showing the same gene signature, suggesting indeed that these endothelial cells in these co-opted tumors are largely quiescent, and thus not a target for anti-vessel cooption therapy, probably. We then thought, well, maybe we need to look at the parasites, and indeed, in the untreated tumor at day 21, you see that there are what we call angiogenic parasites, because they express a lot of genes involved in maturation and sprouting and matrix remodeling, which is completely different for the tumors that grow via vessel cooption, where now the parasites actually at day 21 or 36 sunitinib treatment resemble actually the gene signatures in the healthy lung. And you see indeed that in the before sunitinib treat, you have angiogenic parasites, and later on you have quiescent parasites. So again, co-opted parasites are probably not a good target for anti-vessel cooption therapy. So do immune and/or cancer cells contribute to vessel cooption then? Their role has actually never been studied. And indeed, we see in the vessel co-opted tumors, we see that there are more matrix remodeling macrophages, which by immunostaining are strategically positioned at the invasive front and may assist the invading cancer cells at the invasive front to actually invade into the healthy tissue. So raising the question whether these co-opting macrophages may be a new target. We also considered the cancer cells and find that co-opting cancer cells are in fact more invasive, and that hypoxic cancer cells are also more invasive, again raising the question whether co-opting cancer cells may be a new target for anti-vessel cooption therapy. So in lung metastasis growing via angiogenesis, we see sprouting blood vessels with angiogenic endothelial cells and parasites. We see active cancer cells and immunosuppressive anti-inflammatory macrophages. In lung metastasis growing via vessel cooption, we see co-opted blood vessels, but there the endothelial cells and the parasites are quiescent. And there are, in contrast, co-opting invasive cancer cells and matrix remodeling macrophages of the M1-like macrophages, which may become considered as new targets. Now, how heterogeneous are healthy endothelial cells across tissue? And do we need to consider this for tissue type-specific anti-angiogenic therapy? So we generated an endothelial cell atlas and of 11 tissues, more than 32,000 cells. And what we found is curiously that endothelial cells from certain tissues, like for instance, testis and brain, lung and spleen, seem to cluster together, arguing that they're more similar to each other than to others. And this probably relates to the fact that they share similar biological properties. For instance, the intestines are involved in a vascular gut barrier, spleen and liver in scavenging, brain and testes in barrier, and so on. And what is striking is that arterial endothelial cells actually share many common tissue-specific markers together with each other, whereas capillaries don't, and actually, as you can see in blue, express many more tissue-specific capillary markers. And this probably relates to the fact that the capillaries serve tissue-type-specific needs. We also see that endothelial cells in vascular beds transcriptomically resemble each other across tissues. For instance, arteries cluster together, capillaries cluster together across tissues. And not the position in the vascular tree, but the tissue type seems to determine the endothelial cell heterogeneity. So this may have implications for tissue type specific anti-angiogenic therapy. And as I showed you already, you can use in silico lineage tracing to actually molecularly reconstruct the vascular tree. It's going from, in the left here, the large arteries, and then capillaries, arterioles, capillaries, venules, and then veins. And for each of those, you can actually find and identify a molecular fingerprint pattern. Now, when we look at the metabolic gene signature, then we see actually the same type of clustering as we saw when we analyzed all the genes. So, for instance, the brain and the testis cluster together, the colon and the small intestine, etc. This indicates that actually the metabolic genes contribute to a large extent to this type of clustering. Certain endothelial cells, for instance, in spleen, are more involved in cholesterol metabolism, others in the brain in transporters. So there are tissue-specific metabolic gene signatures for these endothelial cells. And there is also tissue-specific heterogeneity of metabolic gene signature of endothelial cells within vascular beds, which seems to be even different between tissues. For instance, in the liver, you have a metabolic marker for an arterial endothelial cell, another for a capillary, and another for the venous endothelial cell. And whereas this is not the case in brain endothelial cells. Now we're making a mega single-cell endothelial cell atlas comprising 124 studies, more than 15 million cells, more than 700,000 endothelial cells, with across development and adult studies, with more than 40 studies and 30 conditions. And just to increase your appetite, here is what we see for the lung endothelial cells at this stage, where it becomes clear that scavengers, so these immunomodulatory endothelial cells, actually play an important role and make up a large fraction. And systemic venous endothelial cells also seem to be quite predominant in the lung circulation. Finally, I would like to end with our efforts to bridge the valley of death, because what we want to do is find solutions for bridging the valley of death. Translating academic research into drug development is really a big challenge. And we want to do this by improving target discovery. The human genome contains 20,000 genes, but today still 6,000 of them are mystery genes, which lack any annotated function. And to demystify these mystery genes, we built an artificial intelligence tool which identifies genes with a similar role as training seed genes with a known role. So it's guilt by association. And we focus on immunosuppressive genes in endothelial cells. But at the same time, we also build new tools for improved target validation. And in fact, what we developed is a new type of lipid nanoparticles that are selectively taken up by endothelial cells. And if you include into these lipid nanoparticles single guide RNAs, and you inject them into a mouse that selectively expresses Cas9 into endothelial cells, then you can actually obtain an endothelial cell selective knockout in a matter of days and with only a few €100 per cost. We also use this technology for lipid nanoparticle siRNA-mediated silencing. So we want to use these methods for improving alternative immunotherapy to overcome resistance to traditional immunotherapy, which classically targets immune cells and cancer cells. And this resistance is due to the fact that stimulatory immune cells fail to infiltrate, while suppressive immune cells do infiltrate into cancer microenvironment. And we believe and hypothesize that the tumor endothelial cells, these immunomodulatory endothelial cells, are much more important in creating an immunosuppressive microenvironment, and therefore want actually to silence those so that we can make these IMEX more immunostimulatory and increase or overcome the resistance to current immunotherapy. Here is the evidence that the AI tool correctly predicts mystery genes in 95% of the cases. And if we look at the rank plot of predicted genes, then we find on the right, on the top, in red training genes, as expected, because they are the most similar to themselves. But we also find mystery genes in green, which indicates that they are similar to the training genes. And in this case, we used training genes involved in tip cells. And what you see here is also that there is a new mystery gene that we identified. And this really is involved in angiogenesis, because if you silence this mystery gene, then you see reduced vessel sprouting as well as ocular angiogenesis in vivo. We also validated the lipid nanoparticle strategy and therefore focused on the pro-angiogenic VEGF receptor 2, because it's known that when you block it, that then reduces tumor burden and tumor vessels. And so when we inject these VEGF receptor SIRNAs and lipid nanoparticles into tumor-bearing mice, then you see that the expression in red is really reduced. But in panel B and C, you see also that tumor burden and tumor vasculature, tumor angiogenesis is seriously reduced, as you expect. So this is a positive control. We also use this artificial intelligence LNP revolt technology to discover and validate mystery immunosuppressive genes. For instance, in this case, there's another gene that is highly expressed in the tumor endothelial cell. It's a mystery gene, and when you silence it, you use the LNPs, you see that tumor growth is reduced and that natural killer cells accumulate, which may contribute to the phenotype. So with this technology, we can do target discovery and validation at an unprecedented scale, and we start thinking about RNA medicine, whereby you can benefit from that, for instance, combination therapy, etc. So we'd like to thank you and acknowledge also all the people who have contributed to these studies, and would like to answer and discuss questions with you later. Thank you very much.
Well, we're back in the studio here at Houston Methodist Hospital. You just heard from Dr. Peter Carmelit, who has really done some spectacular work in angiogenesis, and we learned a lot of new things today, Peter. We already have questions coming in from the audience. I just want to remind everybody that you can join us here in the studio. You can join by text, and simply text DeBakey to 37607 and then text in your question. There's an alternate approach. You can join by web. You can go to pollev, that's POLLEV.com, and enter DeBakey, DEBAKEY, and then respond to the activity. Well, Peter, that was just marvelous, and I'm going to let my junior faculty member, Dr. Li Lai, have the first question.
Thank you so much for the talk. It's very, very informative, and we certainly enjoyed your insights in vascular metabolism and heterogeneity. So thank you so much for developing those bioinformatic tools for us to use, and I'm particularly interested in the tool called Endogen. Can you introduce a little bit more in detail about the principle of this tool?
It's basically a computational model, where you include and input, let's say, all the metabolically active enzymes and steps, and you then tailor it to the endothelial cells, because every cell type has its own metabolism. They all express or can express the same metabolic genes, the 2,000, 3,000 metabolic genes, but their activity will be different in different cell types. And so what we do is we use single cell omics data and functional data from endothelial cells in vitro and in vivo to then tailor it to an endothelial cell genome scale model. So which is different from a liver cell genome scale model. And so what you then can do is basically silence in silico each of these 2,000-3,000 metabolic genes alone or in combination, and you can do it in a graded manner as well. And then you can find out, does it affect proliferation, collagen synthesis, survival, etc. And so you then get a rank of those genes, those metabolic genes, that are most important, that are most essential for this particular process. So it gives you actually, it's sort of an alternative for measuring the flux activity. It gives you a functional readout, a prediction, obviously, for the functional readout of, let's say, the single cell transcriptomic data, where you can always argue, yeah, but the transcript levels don't match or correspond to flux activity metabolic levels.
Dr. Lai,
so that's the power.
We have, Dr. Lai, we have a lot of questions coming in from the audience. Maybe you could go ahead and start with some of those questions.
Yeah. So there's a question. Is the activation of lipid metabolism contribute to energetic robustness of the ECs in a slight analogous way to cardiac metabolism, relying primarily on energy derived from lipids?
So if I understand the question well, so we previously showed that endothelial cells derive more than 85%, 90% of their ATP, the energy, from glycolysis. But fatty acid oxidation is necessary, so for a little amount of energy production, but primarily for sustaining the TCA cycle, for differentiation through epigenetic regulation. I didn't show you, but in quiescent endothelial cells, also for the production of NADPH and reducing agents to maintain and secure oxidative homeostasis. So it's not like in a cardiomyocyte where most of the energy is derived from lipid metabolism.
Okay.
So there's another question. Does the immunomodulatory role of the endothelial cells comprise activation of immune cells to target the host?
So what we see is that the tumor, and especially in the tumor, that the tumor endothelial cells are probably much more immunosuppressive than we realized. And so what they do is they prevent recruitment and the activation and the infiltration of several immune cell types into the tumor. And by turning, let's say, these immunosuppressive into an immunostimulatory activity, we hope that we can actually recruit more positive cancer cell fighting immune cells in the cancer.
Yeah, I have a follow-up question on that. You showed that there's two genes that are involved in the immunosuppressive aspects of the tumor vein endothelial cells. So I think the idea that the cells in the tumor matrix are creating an environment that is immunosuppressive, I think that's well described. But I haven't heard too much conversation about the tumor vein endothelial cells. So I thought that was very interesting. And you described two genes that I haven't really heard playing a role there. Are those genes that you described, CLEC2B and KLRF1, are those potential targets for immunomodulatory drugs in cancer?
Theoretically, yes, but some of these genes also have other effects in other cell types. Mm-hmm. So this needs to be really taken into account. And that's why we favor so much let's say the LNP strategy that I just explained, because that is selective for endothelial cells only. Mm-hmm. And so we have evidence or experience with certain genes that have a known function in, for instance, dendritic cells, but have an opposite function in endothelial cells. If you then selectively target them with the LNPs, then you get the desired therapeutic effect. If you give a small molecule, I don't know what you will get. Probably not a desired therapeutic effect.
And if I could ask one more follow-up question on that, this concept of EC-targeted LNPs, LNPs that are targeting endothelial cells specifically, that's really fascinating and could potentially lead to a lot of novel therapies. Yeah. How do you, is that due to a ligand on the LNP that's covalently linked? No.
No, it's not tagged. It's just a different chemical composition of the lipids. So there are several types of lipids, and we just played around with them, and all at once accidentally found out that there's more selective, well, actually selectively taken up by endothelial cells and primarily in the lungs, the liver, and the spleen. Now we are generating also LNPs that are tagged with an antibody against, for instance, ICAM or V-cadherin or VEGF receptor 2, and hope to achieve, let's say, a broader tissue type targeting activity.
Thanks.
So the next question from audience, can we modulate corresponding endothelial cell proliferation in favor of tissues that are affected by peripheral artery disease?
Yes, I mean, there is plenty of evidence for that. Let's say the gene therapy effects, the recombinant protein therapies from, let's say, 15, 20 years ago with VEGF clearly showed that you can stimulate angiogenesis and neovascularization. The challenge was back then was that VEGF is such a potent angiogenic factor that it was very difficult to dose. And so what you do with gene therapy is you rapidly overdose, and then what you create is sort of an excess angiogenesis, which is poorly structural, poorly functional, sort of a spaghetti type of angiogenesis, which then later regresses. And so does not become connected to the real circulation properly. And what then happens is that these new vessels simply disappear, they regress. But in theory, if we could find alternative angiogenic factors that are not so potent, which you can dose better, and also for prolonged time periods, for instance, like PLGF, as we showed, then you can stimulate neovascularization in ischemic tissues.
Okay. Are the metabolic pathways in endothelial cells that you have found are more adapted to anaerobic regimen and hypoxia?
Yeah, for instance, glycolysis is a good example. I mean, glycolysis actually can continue with very low levels of oxygen, as long as there is some glucose present. Now, the real challenge is then what happens if the endothelial cell has to migrate into the real avascular regions that are too far away, too distant away from the blood vessel that supplies then the glucose. And so in a study that we're still continuing, we've shown that they actually can use gluconeogenic substrates to then actually generate still biomass. So endothelial cells are actually smart cells. They also differ from most other cell types, because many or most other cell types, when you challenge them with low oxygen levels, they will stop growing. Endothelial cells just keep growing at an increased rate. So it was fascinating when we started these endothelial cell metabolism studies. I was really concerned that we would reinvent the wheel. Not reinvent the wheel and just see what others have seen in cancer cells and in other cell types, but that's not at all the case. Endothelial cells have a very specific metabolism, which I think also makes it attractive for therapeutic targeting.
The diapedesis one is good.
Okay. So next question is, does the endothelium formed in cancer have particular feature with regarded to diapedesis?
Diapedesis. The question is about diapedesis in endothelial cells. Is there some feature that of endothelial cells that favors diapedesis in cancer?
I don't remember them from the top of my head, but we have seen in, I believe it was in breast cancer, that these tumor endothelial cells upregulate genes that actually favor this process. Yes.
You know, one of the things that you told us that I thought was fascinating was the tip cells right? I mean, tip cells have a lot of commonality from one cancer to another cancer, from one tissue to another tissue. In fact, from one species to another species, the gene expression. And it seems logical then that if you want to inhibit angiogenesis, you target your therapy at the tip cells. Tell us a little bit about what your thoughts are in terms of therapeutic targeting of tip cells in cancer angiogenesis or plaque neovascularization.
I mean, I would even mention that tip cells are the only phenotype of endothelial cells that you can maintain in culture. So all other phenotypes that we see in vivo with single cell omics analysis, you lose after 24 to 48 hours. So the dip cell phenotype is extremely robust. And yes, I mean, theoretically, this is one of the ideal target cells. The problem is only that there are so few in human cancer. In mouse cancer, you would see 30% of the endothelial cells are dip cells. In human cancer, it's 10% or less, which might be the reason why, for instance, a drug like anti-VEGF does not work better than hoped. But in theory, maybe it's worth trying. But I mean, the surprise to me was that actually the field has focused for decades on what we call pruning the vasculature. And now you see that you can only prune 10% of the tumor vasculature. And what are all the 90% of the other tumor endothelial cells doing? I mean, this made us think that we should actually re-phenotype and retune these other 90% of tumor endothelial cells into phenotypes that would normalize the vasculature, that would make them more immunostimulatory, etc., etc. And with plaques, we even know less about this. There is also evidence that in atherosclerotic plaques, I think it is in mouse, and that is stimulated by disturbed flow, that also lead to an immunomodulatory phenotype. So we see actually these IMEC phenotypes, these immunomodulatory endothelial cell phenotypes, we see them appear in many diseases, pulmonary hypertension, other diseases, etc. And it's probably a reversible state rather than a real cell type. But which is amenable, or which can be targeted.
You've just on that topic of endothelial cell heterogeneity, you've shown us beautiful work, including your mega atlas on endothelial cells. Have you combined lineage tracing with those data? And the reason I ask that is Lily's got some beautiful data suggesting that in vivo there is an important contribution to angiogenesis of transdifferentiation of a set of fibroblasts to endothelial cells. What do you think about that and any lineage tracing studies?
No, we have personally not done any, let's say, the classical genetic lineage tracing. We only have done in silico lineage tracing using pseudotime analysis, etc. And I think, I mean, endothelial cells are very well known to be extremely plastic. They can transdifferentiate to parasites, to fibroblasts, to hematopoietic stem cells, to immune cells, etc., etc. And vice versa, certain fibroblast types can also transdifferentiate to endothelial cells. So to me, this makes a lot of sense. The vasculature is well adapted to actually, or well suited to adapt to different conditions to fulfill the needs.
Mm-hmm. Well, I'll let my colleague, Dr. Lilai, ask the last question.
So the last question is going to be, are there any endothelial phenotypes that emanate from changes in parenchymal cancer cells?
You mean the cancer cells themselves?
Yeah, I think that's so right. I think the parenchyma of the cancer cells, like glioblastoma, for example.
Yeah. So there are a few studies documenting that cancer cells can actually transdifferentiate to endothelial cells. It's a process called vascular mimicry. We have not seen any of that in our single cell studies, and it's probably because this is cancer type specific. The molecular mechanisms are actually poorly studied, and really not much is known. Yeah. I just wanted to add one more thing. I noticed that one critical slide somehow disappeared, and that's about our efforts to start, or we have started a clinical trial, phase 2 clinical trial for the treatment of lymphedema patients with a ketogenic diet. Perfect. Based on our mouse studies. And there was also one other slide that disappeared. So we have used a ketogenic diet to treat a mouse model of lymphedema, and there we see a beneficial effect, and that formed the basis actually for starting this clinical trial. Which, I mean, we never will get rich of it because everyone can buy ketogenic diet on the internet. But it's sort of appealing that first of all, this was 9 years after we started the first studies on endothelial cell metabolism. And I think it also is relevant for your first question. I mean, are these insights relevant for, let's say, stimulating angiogenesis or neovascularization in ischemic conditions? Here, in lymphedema, we try to stimulate lymphatic endothelial cells, not to inhibit endothelial cells like we do in a tumor. And this is proof of evidence that actually there are ways to do it. So it's worth further continuation, I think.
Really spectacular science, Peter, and with clinical ramifications, and I really do appreciate you being on the show today. Thank you so much.
Thank you very much for the opportunity. I enjoyed it. Thank you.
Thanks to you, Dr. Lee Lai, for joining me here in the studio, and our audience for joining us at the cutting edge of cardiovascular sciences.
Okay, thank you very much. Have a nice day. Bye-bye.